GenAmplify Corona Virus Disease-2019 rRT-PCR Detection Kit Tutorial Series. COVID-19 rRT-PCR assay of extracted viral RNA. Follow proper donning of PPEs. Before pre-amplification, decontaminate the BSC area following this protocol. Decontaminate surface with 10% bleach from center going outwards. Decontaminate pipetors with 10% bleach from tip to handle in a single direction. Decontaminate tip boxes with 10% bleach from mouth backwards. Decontaminate surface with 70% ethanol from center going outwards. Decontaminate pipetors with 70% ethanol from tip to handle in a single direction. Decontaminate tip boxes with 70% ethanol from mouth backwards. Change gloves before preparing reagents to be used. Thaw all reagents on ice. Prepare the number of tubes required to perform the test. Refer to the table shown when running one sample. Note that the master mix contains light-sensitive reagents. Therefore, avoid its exposure to light. Mix thoroughly all the components in all the PCR tubes and spin down or centrifuge 30 to 60 seconds before putting in the PCR machine. Turn on the real-time PCR machine and allow the block to reach optimal temperature. Perform plate setup and select cycling protocol on the instrument based on the protocol shown. After completion of the run, save and analyze the data following the instrument manufacturer's instructions. Analysis should be performed separately for each target using a manual threshold setting. Thresholds should be adjusted to fall within the exponential phase of the fluorescence curves and above any background signal. The procedure chosen for setting the threshold should be used consistently. Interpretation and reporting of results. Results generated by the test can be based on the following. No template controls, or NTCs, should be negative and should not exhibit fluorescence growth curves that cross the threshold line. Positive control or PC reaction should produce a positive result with an expected CT value. Internal amplification control or IAC should be positive at or before 38 cycles for all clinical samples, thus indicating the presence of sufficient nucleic acid from human RNA's P gene. Brought to you by Manila Health Tech Incorporated, Technology Simplified.